Imagine a world where the very building blocks of your life, your DNA, are slowly crumbling, leading to devastating illnesses. This is the reality for some, and groundbreaking new research from the University of Wisconsin-Madison is shedding light on a critical cause: malfunctioning protein that stabilizes our chromosomes. This discovery, published in the prestigious journal Science, could revolutionize how we diagnose and potentially treat certain cancers and bone marrow diseases. But here's where it gets controversial. It challenges some long-held assumptions about how our bodies protect our genetic information. Let's break it down. Think of your chromosomes as instruction manuals containing all the information needed to build and maintain your body. These manuals are made of DNA and proteins, and they reside inside every cell. To protect these precious instructions from damage, each chromosome has protective caps called telomeres. Imagine the plastic tips on shoelaces, preventing them from fraying. Telomeres are made of repetitive DNA sequences and proteins. As we age, these telomeres naturally shorten. However, when the process of telomere formation and maintenance goes wrong, our DNA becomes unstable, leading to premature aging and a host of serious diseases. This is where the new research comes in. Professor C.I.G. Lim and his team at UW-Madison, in collaboration with the university's Department of Chemistry, were on a mission to identify proteins that interact with telomerase, an enzyme crucially responsible for maintaining telomeres. The idea is simple: if telomerase isn't working properly, it could be due to a problem with the proteins it interacts with. Malfunctions in these proteins could be the hidden cause of diseases linked to shortened telomeres. This line of research goes beyond a biochemical understanding of a molecular process. It deepens clinical understanding of telomere diseases, says Lim, highlighting the direct impact of this work on patient care. The research team, spearheaded by graduate student Saurav Agrawal, research scientist Xiaohua Lin, and postdoctoral researcher Vivek Verka, employed a cutting-edge machine learning tool called AlphaFold. AlphaFold predicts the 3D structure of proteins and how they interact with each other. Using AlphaFold, they discovered that a molecule called replication protein A, or RPA, plays a vital and previously underappreciated role, stimulating telomerase, and, therefore, maintaining telomeres. And this is the part most people miss. RPA's primary function was always thought to be DNA replication and repair. Its involvement in telomere maintenance was largely unknown. The team then experimentally validated their findings, confirming that RPA is indeed essential for stimulating telomerase and maintaining long, healthy telomeres in humans. This is a major breakthrough because it provides a concrete explanation for some previously unexplained cases of telomere-related diseases. Lim states that their findings have immediate implications for patients suffering from often fatal illnesses resulting from shortened telomeres, such as aplastic anemia, myelodysplastic syndrome, and acute myeloid leukemia. According to Lim, there are some patients with shortened telomere disorders that couldn't be explained with our previous body of knowledge. Now we have an answer to the underlying cause of some of these short telomere disease mutations. It is a result of RPA not being able to stimulate telomerase. What does this mean in practice? It means that doctors now have a new avenue to explore when diagnosing these complex diseases. The impact of this research is already being felt globally. Lim and his team have received inquiries from clinicians and scientists worldwide, eager to investigate whether their patients' conditions could be linked to genetic mutations affecting RPA's newfound function. There are colleagues reaching out from France, Israel, and Australia. They just want to give a cause for their patients' short telomere disease so that the patients and their families can understand what is happening and why. With biochemical analysis, we can test their patient's mutation to see if it impacts how RPA interacts with telomerase, and give the doctors insights into possible causes of their patients' diseases, explains Lim. This raises a critical question. Could this discovery pave the way for new therapies targeting RPA to combat these devastating diseases? It's a promising avenue for future research. But here's another thought to consider. If RPA is so crucial for both DNA replication and telomere maintenance, could interventions targeting RPA have unintended consequences? This is where ethical considerations come into play. Could enhancing RPA function in telomere maintenance, for example, negatively impact DNA replication in other parts of the body? What are your thoughts on this? Do you think the potential benefits of targeting RPA outweigh the risks? Share your opinions in the comments below.